In a study recently published on the medRxiv preprint server, researchers showed that plasma levels of leucine-rich repeat 15 are associated with severe coronavirus disease 2019 in patients with advanced end-stage renal disease, strong predictor of degree. A population-based study estimated the hazard ratio for ESKD and COVID-19 to be 3.69. Similarly, a European registry study reported 23.9% mortality in dialysis patients with COVID-19. Some of these studies suggest that patients with end-stage renal disease are at increased risk for progression to severe disease. Unfortunately, patients with end-stage renal disease also have an impaired response to the COVID-19 vaccine, especially those receiving hemodialysis. Because these patients regularly visit healthcare facilities for dialysis, they provide a unique opportunity for continuous blood draw for outpatients and inpatients infected with severe acute respiratory syndrome coronavirus 2. About research. In the current longitudinal analysis, we recruited ESKD hemodialysis patients with COVID-19 to avoid selection bias inherent in studies limited to hospitalized patients. The team collected blood samples from ESKD patients at 3 time points: the pre-infection phase, consecutively during SARS-CoV-2 infection, and after full clinical recovery. They performed plasma proteomics, RNA sequencing, and flow cytometry of cytokines and soluble receptors and circulating proteins of immune cells, especially peripheral blood mononuclear cells, to gain insight into host responses to SARS response to COVID-2. The researchers used the Aptamer-based SomaScan platform to cover the plasma proteome of 6,323 proteins as broadly as possible. They link proteomic and transcriptomic data and cellular changes to circulating proteins and provide a comprehensive overview of the multi-source structure of COVID-19. Additionally, the team used supervised learning to identify plasma levels of the LRRC15 protein, a recently proposed surrogate receptor for SARS-CoV-2 and an important biomarker of COVID-19 severity. Research result: PBMC transcriptome analysis identified many upregulated signaling pathways in ESKD patients with COVID-19, such as B type 1 interferon signaling, genes reflecting leukocyte vascular interactions, and the complement cascade. Other upregulated signaling pathways include polo-like kinase mediated events critical for promoting growth stage 2 and mitotic transition, GOLG.